Hello, and welcome back to the Intro to BioCyc webinar series. I'm Alexander Shearer, speaking for SRI's Bioinformatics Research Group. Last time, we took a look at what a pathway page looks like, using the example of the threonine biosynthesis pathway in EcoCyc. This time, we're going to take a look at a protein. In particular, let's try taking a closer look at THRA, and I can do that by clicking through from the pathway page. Here we are on an example protein page from a BioCyc database. Now, there's a couple things we can tell right away. One, this is a well-curated protein in terms of having a lot of information, just typical for something like EcoCyc or MetaCyc. Two, it's a bifunctional enzyme. You can tell when we look at the name at the top where there's name 1 / name 2. And so I picked a bifunctional enzyme on purpose to show a little bit later of the gene reaction schematic. But let's start from the top and see what's actually in a protein page. As I just said, we have the name, and here it's a bifunctional enzyme. Next, we have a button where we click for the protein sequence, just like we did before from a gene page. Following that, we have a series of synonyms, and then below that, a summary. That's a free text description of what the protein does, its role in the cell, and so forth. And this really highlights our assertion citation model for how we write these summaries, where we say that it does something or that it is something, and then we have the supporting citations that you can click through and read for yourself. For example, when I say here, the homoserine dehydrogenase activity of THRA is inhibited by direct binding of substrates for the kinase activity within the dehydrogenase domain, I'm then supporting it with information from this 1977 paper from Wright and Takahashi. And you can click through to that paper, as we showed before. Similarly, you may sometimes see an evidence code up here for a non-enzymatic protein, and otherwise you're going to see some evidence codes up ahead that I'll talk about a little bit later. Following the summary, we have a link to the gene that codes for the protein, a structural description. This is a homotetramer, so we tell you that. The sequence length, a calculated molecular weight from the nucleotide sequence, and any experimental molecular weight data, in this case from a 1978 paper. Down below, scrolling down, we have unification links. These are links to other databases like we saw on the gene page before. In this case, we have links to the E. coli wiki page for this protein, Modbase, RefSeq, and UniProt. You'll also find links to protein structures when those have been determined. Then we have the gene reaction schematic. Again, you see the question mark in the green space. That goes through to a description. The gene reaction schematic is a quick, at-a-glance view of how a gene, a protein, and a reaction relate, or in this case, how multiple genes, proteins, and reactions relate. We start at the right with a gene. In this case, we have THRA, Lys-C, and Met-L. Then they go over to the protein they code for. Then if that protein multimerizes, then any reactions that protein catalyzes. So in this case, we can see at a glance that THRA turns into a homotetramer and carries out two different reactions, which are also carried out by two other multimeric proteins. And that's handy. And you can click through all this. So say I want to click through to the protein Lys-C. Now here we are on the page for Lys-C, or aspartate kinase 3. And again, down at the bottom, we have a gene reaction schematic, and we can click back through to THRA. This lets you quickly navigate between these proteins and the reactions they catalyze, and you can compare and contrast the different proteins that catalyze the same reactions. Below the gene reaction schematic, we have Go terms. Go terms are actually assigned in pathway tools at the protein level and then propagated to the appropriate genes. This makes it easier for things like multi-component enzymes and other similar systems. Once again, we have a Go term, we have an evidence code, just like the evidence codes we're assigning all over the place elsewhere, and a literature citation. Down below that, we have multi-fun terms. Again, we have a last curated tag. Again, it's me. And then down below that, we have enzymatic reactions. So an enzymatic reaction region describes how a particular enzyme catalyzes a particular reaction. Now, it's important to understand that there are separate reaction pages, and the protein page is actually structurally separate in the database. This describes the specific details around one enzyme's activity in one reaction. So let me tell you what I mean. The enzymatic reaction area captures things like, well, the identity of the reaction, which is a different object, but then the KMs for the different components of that reaction, for example, or the known inhibitors and how they work, competitive, unknown, activators, and it also has a summary space for things that we currently don't encode in a formal manner, like in this case, the Ki for threonine. Over here we also have an evidence code that relates to this specific enzymatic reaction, and this tells us that an assay of purified protein was used to let us know that THRA can actually act as an aspartate kinase. And similarly, if we go to the second enzymatic reaction, we learned that an assay of purified protein was also used to show that THRA can act as a homoserine dehydrogenase. And we can see what the KMs and other values are down here for the homoserine dehydrogenase activity. Continuing down the page, we cap out our protein experience with sequence features. The sequence features area of a protein page shows you where different important features, thus the name, map onto the protein. This can be used to show things like different residues that are involved in interacting with an active site ion, a magnesium, other divalent ion. It can show you domains that are involved in binding to a membrane or binding to a substrate. It can show you lots of things. And we actually recently imported a whole bunch of features from Uniprot. So it includes things like this aspartokinase region of interest, sequence conflicts, where Uniprot actually tells us that in the past someone sequenced this and it came out a little bit differently, which is often true with things that have been studied for a while, of course, especially since sequencing has been getting better and better. And other things like this. And so you can look down at the color, find the color on the chart, and see, oh, okay, that's where there's a sequence conflict. Oh, that's where there's a membrane domain. That residue is involved in the enzymatic activity. And again, for each one of these, we'll have an evidence code, and we'll have citation or citations. Well, so that tells us about enzymes. What about reactions? I hope you'll come back for the next part of this webinar series when we take a look at what's in a reaction page in a BioCyc database.